Now, it is my great pleasure to introduce our next speaker, who is our keynote also. He is Professor Manolis Kellis from the Computer Science and Artificial Intelligence Department, a computer science course. He runs the Kellis Lab at the MIT Computer Science and the Broad Institute. He is a true dyed-in-the-wool MIT not creation, perhaps part of our very DNA, if you want, or part of our very structure here. He transcends field boundaries like you wouldn't believe, and does with computers what biologists could only dream of, and oftentimes teaches us things about biology that come from computer science instead of biology proper. The amount of work that he's put out that is actually truly impactful is amazing. Let me just list a couple of things from single-cell genomics, which I think you'll talk to about today, variations in disease, epigenetics, which is a huge, wonderful opportunity right now. We've discussed this with him earlier, and we are both in agreement that olfaction is at the forefront of these things. You can change your olfactory perception by just thinking. You can change your epigenetics by just meditating. And somewhere, the genomics and genetics and epigenetics of all these things connect to olfaction, connect to neuroscience, connect to health and disease, connect to appetite, metabolism, and obesity. So I give you Manolis Kellis, one of the most sort of MIT people you will meet even inside of MIT, and also I believe the youngest ever tenured professor. I think
Maybe not ever, but pretty close.
Well, definitely one of the youngest tenured professors.
How can I plug in?
Please, welcome our speaker.
Thank you. Yes, plugging in would be great.
Okay, you're good.
And then
what do you need?
projector.
What do you have here? This will work. To the
Yes.
Okay, good to know. And relax. You're penultimate. Do whatever you want.
That's awesome. So I am excited to see everyone at the Museum of Science. I don't know if you've told people that we're heading there.
Right, maybe you should announce it that at the end of this, after Shugwang's closing remarks, we will meet at the Museum of Science probably around 6 p.m. or so. We'll give you the exact time. And it will be a semi-guided tour of the nose exhibits there, as well as any other exhibits. Everybody's on their own getting there and on their own getting their tickets. But we will guide you and we will have fun with you. And the museum closes at 9. We should be there at 6. And after that, everybody can go to dinner.
And I'm bringing the kids.
Yes, that's a wonderful thing.
I just realized that my background is always my kids. I can just do this to. So they'll be there, and they hope to see all of you there. And I'm just thrilled to be here. So basically, Andreas stopped by my. Hold on 1 second. I have a phone somewhere.
Whoa, look at it.
He knows to normally do.
I thought Manolis was the goat as a time, but he's actually a goat.
Yes, exactly. Speaking of goats, I was actually kayaking in Greece to this island called, anyway, on the way to Sunion. That's basically about a few kilometers from shore. And as I'm arriving in the midst of these giant waves, I'm basically seeing this completely vertical block of rock. And these black dots on it, they were not trees, they were goats. They were just like at the edge of the cliff. So I'm trying to take a picture of them as the waves are pounding on me, but I don't have quite a clear picture, but that sensation of, wait a minute, these are animals. So anyway, my mom always jokes that she was a goat in a previous lifetime, because every time she goes on a mountain, she's just like on the cliff. So anyway, I feel like I'm here among family. I think Andreas just is the epitome of the sort of next generation scientist of the Renaissance. I feel that every time I speak with him, my brain expands and I'm learning so much more. So he messaged me the other day and he was walking by Stadas. I'm like, hey, do you want to jump on Zoom? He's like, I'm actually downstairs. Where should I find you? And my schedule was packed, his schedule was packed, and somehow we carved out an hour and a half of just brainstorming about olfaction and the brain. And
helped organize our meeting.
so it's really a true pleasure to be here. Thank you for putting together such an amazing program, because I think that, as we heard earlier, this is like the last remaining sense that we need to understand. So I'm going to tell you a little bit about sort of what I do in my day job, which is sort of try to understand disease circuitry. And then I have a few thoughts that I'd love to share just from our sort of short brainstorming session on sort of the connections between deep learning, olfaction, and more generally sort of the future of perception engineering, if you wish. But let me tell you what I do in my day job. So basically what we're trying to do is understand the molecular basis of human disease, understand the mechanism of human disease. So throughout humanity's history, epidemiology has always been about correlation. You know, so to give you an example, the people who can drink Starbucks every day have better health outcomes. Is it because Starbucks coffee is so healthy for you? Or is it because the people who can afford Starbucks every day can also afford better health, better nutrition, better exercise, more time for leisure, better sleep patterns? Certainly not in this crowd. But everything's correlated. And figuring out causation requires two things: either clear temporality or perturbation. And the one place where we can actually get causation out of correlation is actually human genetics. If I inherit a genetic variant and that variant correlates with an outcome, it's very unlikely that the outcome is actually causing that inherited variant that is running through my family. So basically, genetic epidemiology allows us to get a causation. And that's sort of the basis of the open door that we have to understand mechanism. Because everything else is correlation. The one place where we get causation is if we can find genetic differences between individuals that correlate with a phenotypic outcome, and then we figure out the mechanism through which they work, and then we go and perturb that mechanism. That's when we have a dramatically improved chance of actually having a therapeutic that works. Because all of these studies that have basically looked at curing mice from every ailment you can imagine, we have the world's healthiest mice, you could possibly believe in science. Every single time we try to take that from mouse to humans, there's some kind of back door and pathway where the human system just reacts adversely and sort of, shuts that door. But the moment you have human genetics, then you have a path to actually perturbing that mechanism. So that's what we do in my lab. We basically try to sort of use that window of opportunity through genetics to actually understand mechanism and how to reverse that mechanism. So basically how to enable therapeutics from genomics by both dissecting, understanding, and manipulating disease circuitry at single cell resolution. So that was my talk. Thank you very much. That was great. All right. So what is genetics? Genetics, we've known about genetics forever. Basically, for thousands of years, humans have selectively bred crops to basically make them edible, animals to actually make them, huggable, and wool to make it wearable. So we've also understood inheritance in humans. I have to say the joke here. Basically, when my blonde, blue-eyed, pale son was born, everybody was like, "Oh, where does he get his blue eyes?" And I would always say, "Well, his dad, probably." And of course, my wife eventually sent me the 23andMe comparison of me and my son, and she's like, "You can stop making that joke now." But for a geneticist, the answer of where does he get his blue eyes or blonde hair is obviously both parents, because it's a recessive trait. And that's something that we actually had not understood, the fact that you can have brown-eyed parents for dozens of generations, and then a blue-eyed kid. You don't need to kill your wife. This is just a recessive trait. The first person to truly demonstrate the mechanism through which this happens is Gregor Mendel, through his experiments in peas, by basically showing that dominant and recessive traits can basically lead to this independent assortment, these discrete units of inheritance, these dominant and recessive alleles, and the lack of blending. This was genetics completely worked out in 1866. Gregor Mendel sent his manuscript to Darwin, and 50 years later, when Darwin had died, we found a manuscript on his desk that was still unopened, and it was Gregor Mendel's manuscript. His paper was cited 3 times in 50 years. no one believed that the laws that Mendel had so eloquently dissected in peas actually applied to humans. And the reason is that humans understood about continuous variation, about the fact that if you look at hair color, it's not just like one of three options. It's a continuum. Eye color, it's a continuum. If you look at human height, it's a continuum. So how do you reconcile these continuous phenotypes observed in humans with these completely discrete 1-0 genetic inheritance of Mendel? And that was not done until 50 years after Mendel's discoveries, with Ronald Fisher and other statisticians basically coming in and saying, hey, even 5 Mendelian loci would basically give rise to a semblance of continuous variation. And that's extremely key to realize, that basically it seems continuous, but it could just simply be additive effects of a small number of traits. And in fact, just a week ago, the top of my Google newsfeed, again, it's an adaptive algorithm, so it's maybe my Google newsfeed, is about sort of this comprehensive mapping of human height variation. There are now thousands of loci, every one of which contributes reliably to your height at the scale and magnitude of less than a millimeter. So what am I saying here? I'm basically on one hand telling you, we have causality, we can manipulate the system. On the other hand, I'm telling you that it's not just 5 loci, it's thousands of loci, and not just for human height, but for every single aspect of human biology, of human cognition, of human metabolism, of human olfactory perception. Every aspect of human biology is minutely nudged by thousands of genetic variables, and of course, as you all know, by huge environmental components. So why am I so focused on identifying the genetic circuits underlying these loci? The reason is that when you identify these loci, you have a path to manipulation, and you're not constrained by the minute changes that these genetic variants carry out. You can actually find these circuits and manipulate them a lot more. So basically, just to give you an example, we took the strongest genetic association underlying obesity, and we narrowed it down to a single letter, a T to C alteration. Basically, if you are homozygous for lean predisposition, you have this AT-rich motif. So the vast majority of humans have this AT-rich motif that is bound by this AT-rich interacting domain factor. Okay? The genetic variant is very unusual in that it actually has an impact of 7 pounds in an adult. How can a single nucleotide have an impact of 7 pounds in an adult? It is because it actually rose from 5% frequency to 45% frequency from Africa, in Southeast Asia, in Europe, and so on and so forth. So 42% of European chromosomes actually carry the risk allele. I'm actually homozygous risk for the risk allele. I'm constantly starving and constantly exercising in order to not be obese, because my whole family has that predisposition, and I got 2 alleles from both mom and dad with the risk allele. So what does that do? So basically, if you don't have this AT-rich motif, which is normally bound by ARID5B, that then represses this massive switch. It's not just a little switch on the wall. This is the control panel that we have in the basement for the whole building. So that 12,000 nucleotide enhancer, it's like enormous. Most enhancers are 500 nucleotides. This is 12,000. This 12,000 enhancer is basically at the root of a developmental program that decides if my cells are going to basically store energy as lipids, this is white fat cells, or if they're going to burn energy as heat. And basically this decision is made in the first 3 days of differentiation. Every single time that my fat cells renew, they basically decide if they're going to be fat storing or fat burning cells. And evolution doesn't allow big variations in general. So basically it's very rare that such a strong effect variant rises to high frequency? The reason is probably that in the out of Africa event, when we had to cross deserts and ice ages, etc., humans had to somehow store energy more efficiently. And if you look actually retrospectively, you can actually do retrospective genetics with the Framingham Heart Study that has been basically following individuals across families for multiple generations. You can actually go back to the blood samples from before World War II and look at the correlation between this genetic locus and obesity. And you see no correlation. And after World War II, huge correlation. What happened? Office jobs, McDonald's, fast food, dramatic changes in the environment, and those of us who were doing great and were well suited for storing calories through ice ages are suddenly faced with an abundance of free food and little exercise. So suddenly, that variant that was strong effect and therefore would have been kept at low effect by evolution was tolerated to rise to high enough frequency. And as the environment changed, it became a deleterious variant instead of an advantageous variant. So we are now referring to this as an obesity risk allele, where the risk basically increases your chance of a disease. But in general, we should be thinking of not risk and non-risk because it's all environment dependent. So for example, the sickle cell risk allele is the allele that will keep you alive in Africa in presence of Anopheles and malaria. Basically, the geographic distribution of the sickle cell allele matches exactly the geographic distribution of where malaria is prevalent. And in that environment, it's actually extremely advantageous to be heterozygous for the sickle cell allele because it actually protects your blood from actually carrying out the pathogen. So in the context of this relatively weak effect of 7 pounds, I mean, I can't tell my family, hey, I'm obese because I have 7 extra pounds, but what I can say is that now that I understand the circuitry, I can actually perhaps manipulate that circuitry. And by uncovering this circuitry in this incredible team, I like to joke that we wrote a paper about 1 bit of information in the human genome. So basically, this is the New England Journal of Medicine. As you know, it has an impact factor of something like 80. And we basically wrote about a single letter alteration. But what we did is actually figure out how that letter actually works. And that letter basically is bound, results in this. And I was introduced at a talk by the head of one of the big pharmas in this annual meeting bringing together academia and industry as the author of the most depressing paper of the year. And the reason is that if you look at this genetic association, it basically sits squarely on the FTO gene. There's no doubt that there's only one gene underlying this genetic association. So all of pharma was like thrilled. It's like finally we know the target, because for many of these genetic associations, you have 5 or 6 or 10 genes sitting under this genetic region of association. And in this particular case, there was only one, so people were so excited, and they invested more than $1 billion collectively across the community on understanding the function of this gene. And here we go, and basically infer a circuit in our paper titled FTO obesity variant, where FTO doesn't even appear in the circuitry. What's going on here? What's going on is that this sits right in the middle of the first intron, the non-coding portion. So if you look at the scale here, it's quite daunting for those of you who are not spending your days looking at genomes. Exon 3 is this tiny little dot. Exon 2 is this tiny little dot. Exon 1 is, beyond the edge of the slide. So 1.5% of the human genome codes for protein. We basically have 100, centimeters, and one of them is going to be, the exons, sprinkled among very large non-coding regions. And in this particular case, the genetic association basically spans 89 common variants. Just to give you a little bit of background, so we might look like a population of 6 billion people. We're an effective population size of 10,000 people. Basically, human genetic variation was fine and well in Africa, and that's where most of genetic variation still lies, but a very small group of people went off and then populated the rest of the planet. So if you look at our effective population size, which is a measure of the diversity of humanity, basically how many mutations have we accumulated, that dramatic population growth doesn't count. We are basically still a very small population. And if you look at the number of independent genetic alleles, independent blocks, then what you see is that from mother to son and from mother to daughter and from father to son and father to daughter, you basically have blocks of co-inheritance. So you don't just inherit that one nucleotide, you inherit a whole block of 50,000 nucleotides with all of the variation that sits within that block. Everybody with me here? Mm-hmm. So there are 89 common variants that are co-inherited in this locus, only one of which is in fact the driver, as we showed here. And to go back to why this story is so depressing, that variant that basically sits in the middle of this gene, in fact, doesn't control this gene at all. It controls two different genes that are sitting 1.2 million nucleotides away. So at the scale of this picture, this is basically beyond Harvard. This is, I mean, this is extremely, extremely far away. So that basically means we can't just look under the lamppost. And that also means that we need to understand the circuitry of the human genome. We need to understand in every region of genetic association, what are the nucleotides that are co-inherited, that are associated with that disease? What are the regulatory control regions where these nucleotides are found? What are the regulatory motifs that these nucleotides disrupt? What are the regulators that bind these letters, these nucleotides? What are the tissues and cell types where these control regions are active? And ultimately, what are the target genes through which the biological effect happens? Is everybody with me here? Awesome. So that's what we need to do for every one of 120,000 genetic associations. Okay? So basically, in the time of Mendel, he had like a few dozen traits, and that was nice and good, and they were all independent, except for a couple where he had to possibly fudge the data to actually make them look independent. But what Morgan and Sturtevant actually discovered is that the slight, quote-unquote, inaccuracies, the slight deviations from the law of independent assortment that Gregor Mendel had discovered or had inferred from his data, these slight deviations were in fact the basis for genetic mapping. Because if two genes, which we now call genes, are segregating somewhat dependently, then that means that they're close to each other on the chromosome. And that's how dozens of Mendelian loci were mapped in the 80s, by basically tiling the genome with markers and then looking in large families of who inherited which version of a trait, and then mapping the trait inheritance pattern with the marker inheritance pattern, thereby leading to that colocalization and ultimately finding the genes for a bunch of traits. With complex traits that basically by their nature have thousands of underlying loci, this is not possible because you cannot track segregation of 1 millimeter differences in height. We vary more between the time we wake up in the morning and the time we go to bed in our height than any one of these thousands of statistically reliably variants allow us to recognize. So how do we deal with all that? We deal with all this by basically understanding that circuitry that I mentioned and then being able to now go in and manipulate things in a way that's much more brutal, quote unquote, than evolution would ever tolerate. So evolution is constantly acting on the human genome. I mean, it is ridiculous to think that somehow humans have escaped evolution. It is constantly acting at every stage of sperm, ovary, zygote, development, There's all of these stages. Why do you not tell your friends that you're pregnant until you're 3 months old? The reason is that there are many embryos that simply don't develop after 3 months. There's massive amounts of checks that constantly happen to basically give rise to the amazingly perfect individuals that I see in this room. I mean, I'm serious. Like, every single one of you is a huge success story of alleles and gametes coming from the parents, and then starting from one cell, leading to thousands of cell divisions, to trillions of cells, with 3 billion letters of DNA functioning amazingly well. It is remarkable. So I want you to maybe call tonight mom and dad and say thank you for making this miracle of life possible for each of us. And the genetic differences that are common in the population have these minute effects. But if you look at what we can do therapeutically, we can basically go beyond what evolution tolerates in the population. And we can actually start manipulating these at much stronger effect. Okay? So the analogy that I like to give is that the volume that, I don't know, your neighbor likes to tolerate is very low. But then the moment you go in the lab, you can just crank it up all the way one way or another way. So basically evolution only tolerates turning these knobs a minute amount. But once we figure out the knobs, we can therapeutically go in and change them a lot more. And that's what we did. We basically said, okay, great. Let's first of all show causality. Let's show that even changing that one letter can actually lead to this dramatic change in cellular phenotype. And indeed, so my cells are unable to thermogenize. They're unable to burn calories as heat, to heat generate. But if you take cells from, basically someone with my genotype, and you CRISPR-Cas9 genome edit the C, which disrupts the AT-rich motif, back into a T that restores the AT-rich motif, then suddenly you're able to restore thermogenesis. You're able to sort of make thermogenesis work again. If you go into mice and you basically say, let's increase or decrease the expression of these downstream target genes that are a million nucleotides away, what you basically see is that the mice lose 50% of their body weight. Because now it's no longer a minute effect. Now you're going in therapeutically and you can do much larger effects. And when you put the mice on a high-fat diet, the mice that are sort of normal genotype gain weight, as anyone would. These mice where this is stuck in the thermogenic position are just burning the calories away when they're awake and when they're sleeping. They're just not changing their appetite. They're not changing their exercise. They're just simply burning calories. So for those of you who want some of those pills, I have them right outside in the back. Just come talk to me. So that's what we want to do for every single one of hundreds of thousands of knobs that we have now found. Everybody with me? So what makes it hard? What makes it hard is that in 93% of cases, these genetic variants are not perturbing genes. They're not perturbing the proteins. What they're perturbing is these sprinkled light switches that are scattered across the human genome. So basically, these hundreds of thousands of genetic associations are extremely difficult to interpret. Okay? So basically, the promise is, woohoo, we have a genetic association. It sits in FTO. Great, let's spend $1 billion. We'll figure it out. Well, we need a path to causality. We need a mechanism. We'll find new target genes, new therapeutics, enable personalized medicine. The challenge is that in 93% of cases, you have this picture, where you have a bunch of variants, not a single one. The target gene is not known. In this case, it was a million nucleotides away, several genes over. The causal variant among all of them is not known. The cell type of action is not known. The relevant pathways are not known, and therefore the mechanism is not known. So you can't intervene. You're stuck. And that's what we're trying to change. How do we change that? By basically understanding how the genetics ultimately acts through every aspect of human biology. So what we need is basically a map of every aspect of human biology. Simple. So we need every cell type across thousands of human cell types, across every individual, across billions of individuals, across every developmental stage, across every disease state, across every perturbation. right? Yes, that's actually what we do. There's no shortcut. Well, except for the fact that if we understand these common variants systematically, then we can bring in machine learning and deep learning and algorithms to understand the rules of how the genome actually works, and then predict that circuitry. Okay? So what we're doing in my lab, despite being a computational lab, we basically burn about $20 million of NIH money every year profiling the heck out of huge cohorts for brain, for heart, for metabolism, for every aspect of human biology that we can get our hands on. Okay? So what we're looking at is human samples from open heart surgery, from epileptic surgery, from muscle biopsies and adipose biopsies, post-mortem samples across every region of the human brain, across thousands of samples, to understand how the RNA changes, that's the expression of the genes that everybody's focused on, and how the epigenome changes. What is the epigenome? The epigenome is how DNA is wrapped around these nucleosomes. We talked about these 147 base pairs, which corresponds to 7 turns for each loop of this DNA helix, the most beautiful, noble molecule of our time, wrapped around these nucleosomes, and how these nucleosomes at roughly 200 nucleotide resolution are basically packaged up the way that you label your boxes when you move. When you move, you basically write living room, kitchen, kid bedroom 1, kid bedroom 2, etc. And when it's time, when you're finally in kid bedroom 2, you open up the kid bedroom 2 boxes. That's exactly how the epigenome works. So you basically have colors, post-translational modifications in the tails of histone proteins that make up nucleosomes, colors, like Post-it notes, that basically say, if you're a liver, open this box. If you're a neuron, open that box. Everybody with me here? So that's what makes a single, talk about an amazing machine. We have a single code, a single automaton, which then self-replicates and then sends signals to each of these cells to differentiate, to package up the stuff that won't be needed, and to open up only the liver regions based, and then all of these form these networks of interconnected neurons, olfactory perception, biological immune functions, and every aspect of human biology, physiology, cognition, etc., is basically enabled by the diversity of collaborating cell types, even though they all magically, I have to say, share the same underlying code. So that's the epigenome. The epigenome is basically this master integrator that basically brings in information from genetics, of course, from disease, of course, but also from the environment, about socioeconomic, chemical, pollutant, age, sex, comorbidities, and of course, all kinds of biological covariates. So basically, this epigenome is this great integrator that we can use to now start understanding how every cell turns that common code into a biological function. Who's with me so far? Yes? Awesome. Who feels that they're learning stuff? Yes? Awesome. Great. I do that in my lecture too, and it's so nice to see thumbs up. This is great. So basically, what we have done in my research group is understood the language of the epigenome. We've basically written machine learning models for systematically understanding what language does the epigenome speak. How does it encode different types of functions? We've discovered distinct signatures for proximal regulatory regions known as promoters, distal regulatory regions known as enhancers, active transcribed regions, repressed regions with a bunch of different modifications. And we've basically done that systematically across not only different tissues of the body, but also different individuals that are all healthy but differ genetically, or I should say and disease individuals that also are modulated by these genetic variants, but also have the disease component perturbing them as well. Everybody with me here? And then ultimately, the reason we generate this data is because we are genomicists. We think big, we think systematically, and we want to integrate all this data computationally to predict the genes, the regions, cell types, the circuits, the mechanisms of action. And once we have those, we've basically established a series of collaborations across every aspect of human biology to start understanding if I now manipulate mesenchymal stem cells in muscle or adipose, or neuronal co-cultures with microglia and astrocytes and vasculature and immune perturbations, then what is that function changing when I perturb the genes that we predict to be drivers? And also in mouse and animal models, how we can do that in the context of complex circuits. Is everybody with me here? Awesome. So I was actually planning to give a talk, not just give an introduction, but I'll just like fly through it. So basically, number one, we build reference epigenomes to predict what are the disease-relevant tissues. Number two, we look at how these epigenomes vary genetically across individuals and with transcription across individuals and in the context of disease. Then go into single cell resolution to understand both epigenomic and transcriptional variation across individuals. Then we look at the cell type specific genetic effects of these variations across single cell, bulk, genome-wide association studies, and quantitative trait loci that modulate expression or methylation, etc. We integrate multiple phenotypes together. We basically look at the convergence of these processes across many tissues, and then we build high throughput ways to manipulate those. So we basically built reference maps of the human epigenome. If you have a phone, you can just scan each of those and sort of look at the different papers. But basically, this was published in a journal issue with like 20 papers from our team that basically sort of painted the largest map of the human epigenome to date, allowing us to now see how genetic variants associated with different traits localized in control regions, in these regulatory elements that are active in different cell types. And that's dramatic. It basically says that if you look at genetic variants associated with height, with blood pressure, with cholesterol, with inflammatory bowel disease, you can actually see that they enrich in enhancers of different tissues. That basically allows this. So Andrew Law, he showed this figure from my paper at a cancer conference, and he says, well, Manolis writes a bunch of papers, but even I can understand this figure. You can read this, and it says height. And then you can read that, and it says human embryonic stem cells. So height variants act in embryonic stem cells. Blood pressure variants act in the left ventricle of the heart. That's where the blood pressure is built up prior to being pumped in the rest of the body. Cholesterol variants act in the liver. And when I say act, what do I mean? We're not even looking at genes here. Genes are completely out of the picture. We're just looking at the epigenome and what regions of this packaging, these little boxes that says living room 1, kid 1, we don't have 2 living rooms, how they act in different tissues. So this is all these circuits, these control regions, these light switches. These are not the light bulbs, they're the light switches on the wall. And it gets super interesting. If you look at fasting glucose-related traits, a mark of type 2 diabetes and type 1 diabetes, you basically see that it's the pancreatic islets where insulin is actually generated. Where if you look at inflammatory bowel disease, inflammatory immune cells, great, check. Bowel disease, digestive tissues, great. If you look at Alzheimer's, well, I expect you guys to put a lot of money on the brain, if you want to guess. And that's where I would have put my money as well, but I would have lost it all. Because it turns out we saw zero enrichment for Alzheimer's and brain. That's a place where you just yell at your postdoc and say, go find that result. No, I'm kidding, you'd never do that. That's the place where you humbly say, well, maybe the books are wrong. Or maybe whole tissue of brain does not capture the subset of cell types in the brain that are the direct impact of genetic variants associated with Alzheimer's. Instead, we found that CD14 plus immune cells that mark both the circulating monocytes in your blood and the resident macrophages in every one of your tissues, including the microglia, the resident immune cells of your brain, are in fact showing a dramatic enrichment. And indeed, we went into those brains and we basically said, well, let's sort nuclei and then select neuronal cells, immune cells, and then glial cells. And what we found is that that enrichment was pointing very specifically to the immune cells that are only making up 5% to 10% of cells in your brain. So that basically says maybe we should be thinking about Alzheimer's as an immune disorder. And when we published that in 2015, like many people said, hey, I told you so, but for once we actually knew what was going on. And when all of these drugs are failing, focusing on the neurons, that's perhaps because that's a downstream effect of something that actually starts much, much earlier. So we've since expanded from 100 epigenomes to 800 epigenomes. We had another paper in Nature just a year and a half ago. Basically, it's here, looking at sort of this, incredible map. And just to be blunt, schizophrenia, neuroticism, etc., they all map to brain. It's not like our brain samples were bogus. No, it's just that excitatory neurons make up the vast majority of the brain. It's also the place where self-reported math ability, intelligence, educational attainment also localize. So I'm sorry again to be so blunt, but yes, there are genetic differences that actually impact the way that your brain works, just like every aspect of human biology. They're having minute effects, but they are immensely reliably found in the biological processes that make sense. By contrast, you see Alzheimer's localizing here in the blood. And, liver with cholesterol, filtration traits with kidney, and so on and so forth. This is also exciting because we can actually take these multifactorial, multifaceted traits and start splitting them up into their parts. Basically say, listen, coronary artery disease affects everything, but if we take the liver subset, then we find very different biological processes than the coronary artery subset, than the adipose subset, and so on and so forth. So we can actually start now mapping these genetic loci to their target genes through these correlations and start linking together for every locus in the genome across these hundreds of thousands of genetic loci, maps that we have made freely available. You can click here and browse through tens of thousands of these pictures that basically tell you how genetic variants are in fact mapping to target genes in specific tissues with specific targets and specific postulated mechanisms of action. So that's what we want to do for every aspect of human biology. And that's looking at reference maps, but we can also do that for these intermediate maps that vary from person to person, to basically look at not just a reference epigenome, but also the epigenome of this person, of every person in this room, which is actually different from each other. And we can look at how these are painting a path of causality from genetic variation to disease. And when this path is extremely large and tiny little effects, these arrows are much larger. And that's very exciting because there's only so many different variants that influence any one enhancer and so many enhancers that influence any one gene and only so many genes that influence any one endophenotype. Is everyone with me here? So basically we're trying to break up this very large path that basically leads to these tiny effects into a smaller path where we can actually find stronger effects. And if you look across 700 individuals from our cohort to look at genetic variation, epigenomic variation, and phenotypic variation, you basically see that if you tell me the genotype of this person at conception, they don't even need to be born, I can tell you what their methylation level in their brain at 93 years of age will look like. This is amazing. It basically says that genetically, for 50,000 loci, I can actually start predicting the epigenome. Why is that exciting? Because instead of doing a genome-wide association study, this very long arrow between genetics and disease, I can basically do smaller arrows. I can first look at genetics to methylation, and I can predict that with a few hundred individuals instead of tens of thousands, because these effects are stronger. I can also look at the bidirectional correlation in a small number of individuals between methylome-wide association studies, how DNA methylation co-varies with disease. No longer causality, because that's a bidirectional arrow. It could be just correlation. Disease could be changing your methylome. But if I start looking not at the observed methylation, but the imputed methylation, the genetically imputed methylation, then I can actually find causality. Because I can predict that before you're even out of the womb. And if that correlates with disease, then I have a unidirectional arrow again. Everybody with me? So this is very powerful. So we can basically do that to find now hundreds of genes associated with Alzheimer's. For every one of those, we have the cell type of action, this is neurons, the directionality, whether it's positively or negatively associated, and a bunch of new loci that were previously not visible because the genetic associations alone have this tiny effect across this very long arrow, but the moment we bridge that gap, we can actually find bigger effects. And of course, instead of doing this at the tissue level, we can now start doing this at the single cell level. So we have basically profiled more than 2,000 human brains, tens of millions of human cells across Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar disorder, Down syndrome, autism, depression, suicide, and the list goes on. And that's just for the brain. And we've looked at multiple regions of the brain, multiple phenotypic measurements of RNA expression, of DNA accessibility, and of course, dozens of cell types, including neurons, astrocytes, oligodendrocytes, microglia, vascular cells, and the list goes on. In the first paper that we also published in Nature a couple of years ago, we basically looked at how early Alzheimer's versus late Alzheimer's versus non-Alzheimer's changes dramatically the expression of dozens of pathways and genes across dozens of cell types. We have since expanded in a paper that was just submitted a week and a half ago at how they change across multiple brain regions, how they change across dozens of different subtypes of excitatory neurons and inhibitory neurons, and how these change across different brain regions. And very importantly, we can take every single cell and start predicting the Alzheimer's-like transcriptome of that cell based on all of the annotations of the individuals, but now applying this at the single cell level. So for every cell, we can say, is it control-like or Alzheimer's-like, and how far down that path it is. And that's exciting because we can now take all of the genes that are known to be genetically associated with Alzheimer's and then see where do they change the most in expression, early, late, or middle. And therefore, when we want to intervene therapeutically, you probably don't want to go here. You want to go here and basically change the root causes of disease rather than the manifestations. And we're able to do that spatially. We're able to now visualize in tissues, in histology samples, the expression of individual cells. You can see here the arteries of, again, open heart surgery, living human hearts, where we can take these sections and basically see the expression of individual genes in individual RNA molecules in the context of their spatial organization. And we're basically using these deep learning transformers to now translate between RNA, the epigenome, imaging, and a huge diversity of phenotypes. We're learning these modules for how these genes are behaving together. We're learning how genetic variants, in fact, impact individual genes in individual tissues. We're able to classify individuals based on their transcriptome across dozens of cell types and predict schizophrenia versus non-schizophrenia individuals. And when we find exceptions, it's always interesting. This person here, this guy, has expression patterns that scream out schizophrenia, but he was not diagnosed. Instead, his son was actually diagnosed with schizophrenia. What could we do if we had these expression patterns for every person in your family? We could basically say, well, this person was not diagnosed, but genetically and transcriptionally and epigenomically, they appear to be alongside that spectrum of the disease manifestation. We can find the regulators upstream and start giving handles to therapeutic manipulation of large processes involving hundreds of genes, which are targets of these regulators. We can basically look at psychosis, not just in schizophrenia, but psychosis in the context of AD, which is actually associated with this dramatic repression of excitatory and inhibitory neurons, leading to these visions rather than overactivation as one could have naively assumed. We also looked at this single-cell dissection of two different neurodegenerative disorders, a motor disorder and a personality disorder, acting in frontal cortex versus the motor cortex. And basically, we found that, in fact, they are the same disorder, that the same cell type, these long-range projection neurons, which are known as von Economo cells in the frontal cortex and BET cells in the motor neurons, how these are actually altered and along this whole spectrum from ALS to FTD to all of these sort of both tau pathologies and TDP pathologies and A-beta pathologies, we're basically finding these dramatic signatures. So we have a series of success story after success story. Here's another Nature paper that we published just earlier this year, basically dissecting the human vasculature at single cell resolution. This paper is currently in final stages of review, looking at how that vasculature changes in the context of disease. I had actually an olfaction story that I'm going to squeeze in here, which is basically how do you get this diversity of receptors? The way that you do that is by having every neuron, every olfactory neuron, express only one allele, not even both alleles, only one allele of only one gene. How is that possible? Despite the fact that these olfactory genes are densely clustered. These are all of the olfactory receptors in one of several clusters that you see across the human genome. Epigenomics works in regions. You can repress a region or activate a region. How can you get this unique signature? And what we found in collaboration with another Greek, and in fact two Angelas, so there's Angela and Angeliki, so there's the two devils and the two angels somehow. What we basically found is this amazing signature of this repressive mark, H3K9 trimethyl, and this activating mark, H4K20 trimethyl, which is found specifically in these olfactory receptor clusters. And if you sort all genes in the genome based on that signature, what you basically see is that chemoreceptors are by far at the top of the list. So from the epigenome, we have a distinct signature which actually marks all of these chemical receptors. Again, story after story, I could just go on for a while, but I'm going to tell you one more vignette here of these hallmarks of disease. We can basically take Alzheimer's as a monolithic disease and then wonder why every clinical trial is failing. And the reason is that it's a polyfaceted disease. But what we can do using our thousands of post-mortem human brain samples is actually start looking at what are the hallmarks of disease. How can we now understand Alzheimer's in terms of its molecular basis in the neurons, in excitatory neurons, inhibitory neurons, oligodendrocytes, astrocytes, microglia, and basically recognize the distinct pathways and the distinct paintings of every patient based on those pathways. So we have these transcriptional hallmarks. From those, we can find the distinct patient subgroups, and these actually show distinct diagnosis and prognosis signatures that we can then use to prioritize treatment. So that's the vision of where we want to go. And Andreas mentioned that olfaction, of course, has a huge role in appetite, in eating and exercise, etc. And basically, this is our study showing just a week ago this balance between obesity and exercise, that there are dozens of pathways that are shifted across multiple tissues when you exercise, and dozens that are shifted when you eat high-fat diet. But in fact, the two are actually pushing against each other. And then just very briefly, we've basically developed these modular and programmable constructs that allow us to now start manipulating those. We're developing these extremely high throughput ways of measuring the impacts of those. We are able to measure 7 million enhancer elements in reporter constructs in a single experiment, enabling us to now sort of figure out high resolution marks of what are the nucleotide drivers of enhancer activity across these 500 nucleotide blocks. We can say, no, it's these 5 nucleotides that matter, and pinpoint individual regulatory motifs, individual regions of evolutionary conservation, and we're now able to disrupt those in mouse brains to basically see how they're acting. We've now partnered up with Brad Penteluta and Marika Zidnik. We have our retreat in next Tuesday. What we're basically trying to do is now start synthesizing therapeutics using deep learning methods, understanding all of these incredible wealth of information across both genomic information and also structural information to design new molecules, to synthesize them using click chemistry, using peptide synthesis, and to basically start testing them. So where do we need to go? We need to transform pharma. We need to enable personalized medicine. We need a coalition across computer science, biology, chemistry, biotech, finance, pharma, enabling patients, bringing together hospitals, and basically thinking about disease in a prognosis way, in a pathway-centric way, in a rational way. And the only way to enable personalization is get rid of this false dream of, a pill for every person. Instead, basically think about a pill for every pathway. Because a pill for every person is economically untenable. A pill for every pathway basically means that we can now leverage these sharing of pathways across individuals. And there's only a small number of these common pathways, and we can manipulate them systematically, and every patient will be a combination of pathways based on their burden. So that's where we want to go. We want to basically use deep learning, use sort of this diversity of representation learning across the human cortex, basically start understanding how that architecture that is so pervasive in olfaction is in fact pervasive across every aspect of human cognition, how in fact this encoding of order, light, abstractions, knowledge is actually different and unique in a person-specific way across every person. Understand the diversity of, all of these olfactory neurons that I mentioned earlier, every one of us carries hundreds of olfactory pseudogenes. All of these olfactory genes are constantly getting lost. Why? Because we have tricolor vision. We can tell rotting fruit just by looking at them. We don't have to smell them. And therefore, every person has a unique sense of smell based on the unique subset of chemoreceptors that each of us has, and then, of course, the unique combination. But that's an opportunity to start thinking about multisensorial engineering, to use olfaction as a window to neurodegeneration, as we heard earlier, as a model for distributed perception, as a way to reprogram metabolism, rewiring appetite and sensation with smell recording. Thinking about synesthesia and connections between the senses, between cognition and emotion. Think about a systems level understanding of the biological processes and functions, the epigenomic basis of memory formation, and this multisensorial engineering. So as we start thinking about human-machine interfaces, as we start looking at deep learning and this translation between all of these polyfactorial and polymodal ways of thinking about the world, we need to truly embrace this sort of multi-systems engineering, multidisciplinary approach that, again, our host here embodies so well, and this conference has highlighted so well. I am ridiculously fortunate to work with this unbelievable group of interdisciplinary scientists. So this is our annual retreat in 2021. And we are just thrilled to learn more about this amazing field that you guys are sort of starting to dissect and play out. And I really want to sort of say that this is really a model for human cognition and distributed perception. Every one of us is different, and that's what makes the beauty of humanity, like the joining of all of these brains from just such different perspectives is, I think, the secret to the enlightenment and the current revolution that we're undergoing now. So I'll stop there and let you guys move on. Thank you.